Hello, my name is Josh. I'm here to talk to you about southern blotting. It is my hope through this video that you're able to understand what southern blotting is and how scientists are able to perform it. But before we're able to get started, let's go back down and review a little bit about what cells are in DNA. So you have your person here, and he's made up of millions upon millions of different types of cells, ranging from heart cells to liver cells to skin cells. And if you zoom in on your arm, for example, you're able to see a view of all of these different skin cells. You'll notice that in each and every one of these skin cells is a nucleus. And if you zoom in into the nucleus using a special type of microscope, you're able to see this molecule known as DNA. Now, DNA is extremely important for life because it essentially makes you you. It has all the information needed to make your brain cells, your skin cells, and whatnot. Now, scientists are able to look at this DNA and notice that they have these special types of genes that codes for different things. For example, my DNA right here has a special type of gene called the Bob gene. Now, this Bob gene is used to make a ingredient that is needed to make my hair brown. Now, scientists want to figure out if your DNA also has this Bob gene. They want to figure out if you also have this Bob gene. And they're able to figure this out using the process called southern blotting. Now, in order to do a southern blotting, scientists first need to cut up your DNA. Now, they are going to use a special molecule that's going to cut up the DNA. And some of the cuts are quite small, some of them are actually quite large. So you're going to have like a small cut, a large cut that produces different lengths of DNA. So you're going to be left with these parts of DNA that varies in size. You have large pieces and small pieces, and you're going to cut up your own DNA as well, making large pieces and small pieces and whatnot. Now, these pieces are able to be injected into a special type of gel. Now, this gel is just like Jello. In fact, it looks like a block of Jello, something like this. And the scientists are able to inject it onto this gel into these wells right here. So my DNA goes into this left gel, and your DNA goes into this right gel right here. Now, DNA is unique in that it is negatively charged. And you know in life that when you have a negative particle and a positive particle, they're going to be attracted to each other, while a positive particle and a positive particle are repelled from each other. So things that are alike are repelled, things that are different are attracted to each other. So this DNA is actually negatively charged, and scientists are able to put a positive charge on the bottom of the gel and a negative charge on the top of the gel. And what's going to happen is the DNA pieces are going to want to move towards this positive charge through the gel. Now, this gel is unique in that if you zoom in on it, it actually looks like a spider web. So if you zoom in on it, you see like all of these fibers essentially that make up the gel. Now, the large pieces of DNA are going to get stuck onto the fiber, while the smaller pieces are able to fit through the gaps. So what you're going to have is on your gel, you're going to have all these different pieces that are arranged by size. So you're going to have the larger ones that are stuck up here, while the smaller ones are able to move down further and further onto the gel. Same thing happens with your DNA. You have the larger ones up here and the smaller ones as you move further down onto the cell. The scientists are then able to put a special type of sticky paper on top of your gel. So it goes on top right here. And what's going to happen is that the DNA is going to be pushed up onto the sticky paper. So if you look at it from a side view, you have your gel right here and your sticky paper right here. And your DNA is going to be forced from your gel up onto your sticky paper. It's like a slingshot almost that forces it up onto the sticky paper. So what you're left with is the sticky paper with all of the marks of your DNA left onto it because it got forced onto that sticky paper. Now, scientists know that the Bob gene is attracted to the Sarah gene. So you have your Bob gene right here, and it is attracted to a Sarah gene. So your Bob gene is the green one, the Sarah gene is the blue one, and they're married to each other. So whenever there's a Bob gene, the Sarah gene is going to find it and stick to it. Scientists are able to use this, and they realize that if there is a Sarah gene, it's going to find the Bob gene and stick to it. So scientists put this special molecule onto the Sarah gene and inject a lot of Sarah genes onto this piece of paper. And the Sarah gene is going to look and find the Bob gene and stick to it. And if there's a Bob gene on your DNA, it's also going to find it and stick to it too. And using the special molecule that's on it, it's going to also be there. Now what the scientists are going to do is they're going to put another piece of paper on top of it, and this special piece of paper will interact with the special chemical that was on the serogene and leave a mark onto this extra piece of paper. And what you're going to be left with is a special piece of paper that scientists are able to put underneath a special type of light, and they're able to see if there's a smudge on my DNA and your DNA. And if there's a smudge on your DNA, it proves that your DNA has that Bob gene. So if there's a smudge on yours, that proves that there is a Bob gene on yours. Now this is the process of southern blotting. It essentially proves that your DNA has a gene similar to my DNA. Thank you very much for watching. I hope this helps.